As we move into Chapter 3 in your text, we are going to discuss the foundation information really for the rest of your course. Chapter 3 is titled The Cell, but that really is a little bit misleading. We are going to discuss prokaryotic cells, eukaryotic cells, how they're different. We're going to look at anatomy as well as physiology. So this is really a much bigger chapter than just those two title words will lead you to believe. This chapter really begins with a look at some of the important people in the history of microbiology. Now, most of this history that we're going to go through really relates back to the theory of spontaneous generation. So you have to think back to before the days of microbiology, before we knew that bacteria existed, before we knew viruses existed. We really had no idea what was causing illness, and for the majority of this time, it was often related to religion and things of that nature. So the theory of spontaneous generation states that life can arise from non-living matter. So to say that a little bit simpler, it means you can take things that aren't alive and then just with um, no magic spark, that life can just form. Well, this is in a way what they were thinking happened with disease. Back before the 1600s, people thought that humans were healthy, and then all of a sudden something bad happened. Maybe something, they didn't know what, but maybe something like a bacteria came to life inside of them and made them ill. So you have your scientists that set forth to really disprove this theory of spontaneous generation. And there's two names that are really important when it comes to this type of work. The first name is Redi, Francesco Redi. Back in the 1600s, he was one of the first scientists to really think spontaneous generation's not real. And he did his experiment that's shown here on the slide. He took raw meat and put it in a test tube, and if he didn't cover it, then maggots would form. Well, we all now know that the maggots were forming because those are the baby flies. So the flies would land on the meat, lay the eggs, and you end up with maggots. So the second experiment that Reddy did, he put a cork, he covered the container, and you got no maggots. So that proved that it had something to do with the flies landing on the meat. But Reddy didn't really understand what. So some critics then told Reddy, well, it's because you didn't let any air get in. You have to have air for life to magically occur. So Reddy did his experiment again, and he put some gauze on the top, and he showed that the maggots actually formed on the gauze. Air could still get to the meat, but the flies couldn't, so no maggots on the meat. So this was a pretty good experiment, but he's really not credited with completely disproving spontaneous generation. The person that gets the credit for disproving spontaneous generation in 1862 is Louis Pasteur. Now, we'll learn as we go through that Pasteur also invented pasteurization, several other things, but this is really, as microbiologists would consider, his most important experiment. And I covered this experiment a little bit in Chapter 1. So with this experiment, Pasteur took a flask with a curved neck, and he heated up the liquid inside of the flask. When he let it cool, no contamination. What we now completely understand is the bacteria from the air comes in, gets trapped in the neck of the flask. He then can break that flask off, let this sit exposed to air, and because there's no curved neck to catch whatever's in the air, those organisms in the air, they get directly into the liquid in the flask, contamination occurs. So to me, if you step back and look at it, these experiments are almost identical. I do think that Pasteur is really known as the most important person for disproving spontaneous generation because his experiments occurred later in the timeline and people had a more open mind and just understood science and this type of thing a little bit more. So just to go through a few other important people, we have the cell theory to think about. So the cell theory tells us that cells are going to be those fundamental units of life, meaning all living things are composed of some sort of cell. To be a cell, that means that you have compartmentalization, where certain specialization is going to occur within different compartments. Now, all of this in the cell theory started in the 1600s with Robert Hooke. Robert Hooke was not looking at bacteria. Um, Leeuwenhoek was the first guy to look at bacteria, if you recall from chapter 1. Actually, he was looking at eukaryotes, but he was the first one to look at microbes. Robert Hooke was looking at cork, which was non-living, but he could still see the compartments. And then following him, Slyden and Swan also started observing some living tissues, first plants and then animals, and they continued to see these same compartments that we called cells. All right, now our next theory to think about is the germ theory of disease. With the germ theory of disease, this was really thinking about what causes disease. So your first experiments with Pasteur and Reddy, they were looking at something leading to contamination. So growth of some sort of microbe we couldn't see. But then the research kind of shifted some and started going more towards germ theory. They wanted to be able to prove that these small microscopic organisms were the cause of disease. So this actually started, as far as we can see, back with Francestora in the 1500s. He really didn't do any experiments, but he was one of the first people that we can find through literature that had the idea that there was some sort of tiny spore-like items that were being transferred from one person to another, and maybe through direct contact, through the air, something of that nature, and that's what was leading to disease. It wasn't until the 1800s when we really started seeing some evidence for this. First, Semmelweis, he was working with med students, and he started noticing that if med students did an autopsy on a corpse that was sick in some way, meaning what we understand had a bacterial infection, and then went on and did other tests, most commonly vaginal exams on living patients, that if they did not wash their hands in between, I know that sounds terrible, but if they didn't wash their hands in between, the living patient was more likely to die of a disease. So that really set off the trigger of, there's something living inside of that corpse during the autopsy that is being transferred to the living patients. Then a year later, Snow observed people getting cholera, and he related that disease back to sewage contamination in drinking water. I know, again, gross, right? We wouldn't drink water that's sewage contaminated, but in the 1800s, we just really didn't understand. Now, our two most important people that really did a lot with germ theory of disease, Louis Pasteur gets a lot of credit, but it's really Joseph Lister who is the main player in this germ theory of disease. What Pasteur did was he used his previous research with the boiling of the liquid to show that he could make food last longer if he heated it a little bit. And that's where we get pasteurization from. He was really, if you look at it, he was studying more shelf life of food and really beverages, like different alcohols and things like that. What Lister was doing, he was taking Semmelweis's research, and he was really looking at hand washing and taking that further, cleaning surgical instruments. And he was able to start the idea of sterilizing and the aseptic technique that we use in surgery and hospitals and laboratories all over the world today. So Joseph Lister is very important. And you may recognize part of his name from a common item you, I hope, use all the time, Listerine. Listerine was originally created with the chemical that Lister used. It's called phenol. then another important guy in this whole scheme of things with the germ theory of disease is Robert Koch. And he developed the postulates, again, that we went over in chapter 1. So with these postulates, what he was able to show, and I'm going to flip to the picture, and you guys will have the notes to study. He was able to show that if you had a diseased animal, you could look at that blood and see that there's something in there, a pathogen causing the disease. You can then take that particular pathogen, isolate it, which is a fancy way of saying grow it on some media in the lab, then take the isolated pathogen, infect a healthy animal, and then when that animal dies, you can then isolate the pathogen again. So these sets of experiments were showing that it was indeed this microscopic pathogen growing on the media that was causing disease. I pulled this picture directly from your text. I think it's a very easy way for you to study, and not only study to see what each person did, to see their contribution, but it also kind of shows you on a timeline. I'm not going to ask you specific dates and to remember those when you're studying and taking tests, but I think it is nice to see how it all relates together. Now, moving to part 2 of this chapter, enough about the history. Let's look at the actual cells and look at characteristics. So the first thing we need to discuss is what is the difference between a prokaryotic cell and a eukaryotic cell? Well, you probably learned in general biology that eukaryotic cells have a nucleus and prokaryotic cells do not. While that is true, it is much more complex than that. The better sentence is to think about prokaryotic cells do not have complex membrane-bound organelles. So that nucleus is really just an example of an organelle that a eukaryote has that a prokaryote does not. Now, saying that they are different, this slide shows you there are 4 things that are the same or very similar in prokaryotes and eukaryotes. Those 4 things that are similar are the cytoplasm. The cytoplasm is the liquid-like substance that's in the middle of the cell surrounding all the organelles. The plasma membrane, it's always going to be a lipid bilayer, which you learn in general biology. It's shown here, where you have the hydrophilic head groups of the lipids facing out of the cell and facing the inside of the cell. And then you have the hydrophobic tail-like regions in the middle. So that plasma membrane formed from your lipid bilayer creates a layer that does not just easily let things move back and forth. It creates a boundary for your cells. Both prokaryotes and eukaryotes have chromosomes. That's how we carry our genetic material. That's our DNA. So it's living. It has to have that DNA. In order to be living, the cell also has to be able to make proteins. So both prokaryotes and eukaryotes have a ribosome. So now let's look at the prokaryotic cell. This should look very different from the picture of the prokaryotic cell that you've seen before. I want you to really appreciate how complex a prokaryotic cell can be now that we're in microbiology. We're going to see horrific diseases that can be caused by these bacteria. We can't think of them as simple. They are complicated, just in a different way. So what you need to do with this picture here is be able to look at the picture and identify what each of the labeled items looks like and what it does for the cell. Now I have it described on the next slide, but I'm going to go over it while we're looking at the picture together. Okay? So starting from the outside in, the very outside of this particular picture contains a capsule. A capsule is a type of glycocalyx. The glycocalyx is a sugary layer on the outside of your cell. There's 2 types of glycocalyx. The glycocalyx can be slimy, forming a slime layer. The slime layer will help the cell attach to things. This is very common when you think about, for example, bacteria that grow in your teeth. Tomorrow morning, when you wake up, before you brush your teeth, take a fingernail and scrape the front of your teeth. You'll get a nice sticky white substance under your fingernail. That is bacteria sticking together, mainly with that slimy layer of the glycocalyx. Now in this picture, the type of glycocalyx we see is a capsule. The capsule is a much thicker, more hardy, hard layer of a glycocalyx. It's for protection. A lot of times bacteria will use this to protect it from being broken down in our digestive juices or by the whatever environment the bacteria is living in. Beneath the capsule, we have the cell wall. Now we're going to look at the cell wall in a little bit more detail here in just a minute. It's made of peptidoglycan. Underneath the cell wall, we see the plasma membrane. Protruding from the external layers, whether you have the cell wall and a capsule or not, there's options. Some bacteria or prokaryotic cells have these little hair-like extensions called fimbriae. Fimbriae are used for attachment. Sometimes we'll see a long, more flexible extension. This is a flagellum. Now they come in different arrangements. We'll see that in just a minute. A flagellum is used for movement. The third option for a extension is a pilus. A pilus is used to exchange DNA from one prokaryote to another. What the bacteria will do is if they have helpful things in their DNA, they'll put that information in a little extra piece of DNA called a plasmid, shown right here, separate from the rest of the DNA. And then they'll share these advantageous genes from the plasmid. They'll copy that one little piece of DNA and then shoot it from one prokaryote to the next through a pilus. Now looking at the inside of the cell amongst the cytoplasm, as I said, we have ribosomes just floating around. That's where we make proteins. Some prokaryotes have inclusions. This is going to be a storage site. All prokaryotes have a nucleoid. The nucleoid is the concentrated region of the chromosome. It is not a nucleus because it does not have a membrane. So all of that information that I just said to you is kind of written on this slide here, and I've also listed the important page numbers for you. Now looking at a little bit more detail, let's talk about this prokaryotic cell wall. The prokaryotic cell wall is the only cell wall on Earth made of a component called peptidoglycan. Humans don't have a cell wall, but plants, all your other organisms that are eukaryotes have a different type of cell wall. The prokaryotic cell wall, made of peptidoglycan, is very easy to understand. It's made up of layers. The layers have alternating sugars. Okay, so that's what this NAG and NAM. Those are abbreviations for sugars. And alternating means first you have sugar 1, then sugar 2, then sugar 1, sugar 2. So I pulled this picture from the internet just to show you what the sugars look like. You don't have to draw them or anything like that. But what I want to show you is they have the same basic ring structure. So if you kind of hid all this extra junk sticking off the edge, you'd have glucose. But what makes these special sugars are these extra chemical groups hanging off the end. So the NAG, that stands for N-acetyl glucosamine. It's got a nitrogen. The NAM stands for N-acetyl muramic acid. Again, just the different side groups. What makes this cell wall strong is with these layers of alternating sugars, you have connection points. The connections are made by tetrapeptides. That sounds like a fancy word. A tetrapeptide is the fancy way of saying a short protein that is only 4 amino acids long. And so if I flip to the next slide, I think it shows you a little bit better. So in this picture, you've got the little orange and blue looking circles. That's your sugars. So you've got NAG, NAM, NAG, NAM. And then you see connecting all of this, you got 4 little circles. That's representing the 4 amino acids that make up the tetrapeptides. Okay? Now, you notice on this slide, I start using the 2 terms gram-positive and gram-negative. We classify the majority of our prokaryotic cells specifically by the type of cell wall that prokaryotic cell contains. One of the major differences is shown in this picture. Gram-positive, when I think of positive, I think of plus, right? So what does this gram-positive have? What is its plus? What's its extra? In addition to these tetrapeptides, you've also got some pentapeptides. So you just have a little extra connector in there that you don't see with the gram-negative cells. Well, let's look at what else we can see different between them. So I told you gram-positive has extra stuff, right? The main thing we see is with the gram-positive, we've got plus more peptidoglycan. So I like to think of all those P's running together. Positive plus peptidoglycan. And so if we look at this picture down here, you see the blue represents the peptidoglycan, those alternating layers of NAG and NAM. So you see the gram-positive has much more of your peptidoglycan than plasma membrane cytoplasm. So with the cell wall part is just up here. Within these many layers of peptidoglycan, the gram-positive cells have teichoic acids. These are little extra components that make the gram-positive a little bit more rigid. Also does help with binding. So let's look at the gram-negative over here. See the gram-negative, he's minus. He's lacking some of the peptidoglycan. But he does have an extra outer membrane, so a little bit more lipids. And within those lipids, he's got some porins that allow things to move within that extra outer membrane. Okay? So this difference is going to be important for just a couple things throughout the course. First, as I said, we're going to classify our bacteria as gram-positive or gram-negative. So it's very important you understand the difference. In lab, we also use a staining procedure that allows us to see whether the cell is gram-positive or gram-negative. As we get into diseases and how a prokaryote can be pathogenic, we'll also see that there's some things about the gram-positive that help it become a little bit more likely to cause a disease in certain times. Where so your gram-positive is a little bit thicker, so it's a little bit more resistant to mechanical breakage. But with your gram-negative, it's thinner, but it's more resistant to phagocytosis, which is your immune system breaking it down. So we're going to really use this concept of gram-positive and gram-negative quite a bit. Now this slide here just lists the different types of glycocalyx, which I went over previously. Difference between capsule and slime layer. Now let's look at the different ways the prokaryote can arrange flagella. So what this picture shows you is how the flagella really anchors within the cell wall and cell membrane. Now we're not going to learn all of these details, but what I do want you to know is that the flagella really has 3 major parts. It has the basal body. That's this whole region down here that's anchored. Then it has a hook, which forms a right angle. And then attached to the hook is the filament. That's really the long tail-like portion that we see protruding from the prokaryotic cell. Now, it is a little different in gram-positive versus gram-negative because of the thickness of the peptidoglycan cell wall. So what's really different about them is what makes up the basal body. With these prokaryotic flagella, they can be arranged a couple different ways. A prokaryote can have 1 flagella, When it does like this, we call it monotricious. When the prokaryotic cell has two flagella or multiple flagella, but they're always at opposite poles, opposite ends, we call it amphitricious. When the bacteria or prokaryote has a tuft or large collection of prokaryotes in one small region, we call that lophotricious. And if the bacteria is completely covered by flagella, they're all over the surface in random arrangement, we call it peritricious. So it's just important to remember those different types of organization. When we look at this flagellum, we can see how the prokaryote can move. The prokaryotic flagella does not wiggle like a tail in water. The prokaryotic flagella can rotate clockwise or counterclockwise around this hook. So remember, the hook's at a right angle. So it's kind of moving in a lot of ways like a helicopter propeller. If the prokaryote turns all of its flagella in a clockwise movement, that leads to random tumbling. If all the flagella rotate counterclockwise, that makes the prokaryote run in a straight line. So let's think about what the prokaryote would be wanting to get to. And so what's the benefit of this? So let's take phototaxis, for example. What if this is a prokaryotic cell that needs the sunlight? So if it needs sunlight and it's in a dark area, what's it going to do to get to that light? It's going to orient itself towards the light and rotate those flagella counterclockwise to move straight towards the light. Same thing for chemotaxis, responding to chemicals. What if the flagella was trying to get to food? It's going to rotate counterclockwise and move towards food. Well, what if that chemical is Lysol and you've just sprayed Lysol everywhere? Well, that prokaryote is going to be in a negative situation. So negative chemotaxis is going to say, whoa, I just need to get out of the way. So all the flagella will start rotating clockwise, and just random rotation, get that flagella out, get that prokaryote out of harm's way as quick as possible. There are several really good little animated videos you can watch on YouTube, so you can really get a good understanding of the different types of negative and positive photo and chemotaxis. The last structure I want to talk about with the prokaryotic cells is an endospore. Now, there's only 3 different genera, which is plural for genus, that can produce an endospore. Not all prokaryotes can do this, but it's a big advantage to those that can. The prokaryotes that can produce an endospore are the ones we're going to worry about are Bacillus and Clostridium. When we're thinking about an endospore, we'll have a living or what we call vegetative cell. During bad conditions, so say we're out of food, the temperature is not right, there's no water, bad situation, this vegetative cell will begin copying its DNA. It will then sequester the DNA into a small region. That little sequestered DNA will form a forespore, which simply means we start building these very strong protective layers around the DNA. We continue building until we end up forming the endospore. The complete final coating around the endospore that's released contains calcium and dipicolinic acid. It's a very, very strong, almost bony-like substance on the outside of that endospore. So this endospore is now extremely resistant and can live for years and years and years. We honestly don't know how long they can live. We have found endospores in Arctic tundra that were still capable of becoming vegetative cells again. This endospore will just hang out till conditions get good, and then it can germinate forming a brand new, healthy, living vegetative cell. So if you kind of predict and look ahead a little, think about the implications this can have with disease. Would you want to get a disease that when you tried to take medicine to kill it, it could form an endospore that's resistant to the medicine? It sounds kind of scary, right? So one of our last things that we need to discuss with prokaryotic cells are just the overall shapes and arrangements of the cells. Prokaryotic cells can be round. When they're round, we call them coccus, or many of them would be cocci. If they're rod-shaped, or kind of like a grain of rice, we call those bacillus. Plural would be bacilli. If they're shaped like a comma, or curved rod, like this, we call this vibrio. If it's not really a circle, but not really a rod, we call this coccobacillus. Very, very rare. We'll talk about these. Sometimes we see this spiral shape. The spiral shape is termed spirillum. When the spirillum looks like it's been twisted up and it's just really, really tight and twisty, we call those spirochetes. Now we can take each of these shapes and put those together in different arrangements. If we have a single cell, we just use that original term. So looking at these coccus-shaped cells, this one picture would be a coccus. When you have two cocci together, it's a diplococcus. Four cocci together forming a kind of square shape or cube, we would call those a tetrad. A long chain of cocci we call streptococcus. And then a random cluster of cocci, we would call that staphylococcus. So does staphylococcus mean deadly, horrible bacteria? No, it just means bacteria that are round shaped and clustered together. Not all staphylococcus are bad. So you see more of these arrangements with the cocci shape. With the bacillus shape, the only real arrangement you're going to ever see is a streptobacilli, again meaning a chain. So let's finish up the chapter by looking at eukaryotic cells. Now, I don't spend quite as much time on eukaryotic cells because this is the information that you learned in your general biology class. With the eukaryotic cell, we of course have the membrane-bound nucleus holding the DNA. The DNA never leaves the nucleus. Whenever we need to use that DNA to be the code to make a protein, DNA is transcribed to mRNA. The mRNA can leave the nucleus, go into the endoplasmic reticulum. It's called rough endoplasmic reticulum because that's where we see our ribosomes. So that mRNA can be transmitted here and converted into proteins. The proteins then move from the rough ER into the Golgi apparatus. The Golgi is where the proteins are going to be sorted and packaged, maybe even shipped to where they belong. So that's just kind of, I like to describe it that way so you see the path of how everything works together. But there's lots of organelles that are involved here, right? We have mitochondria. Yes, I know that's the powerhouse of the cell, but we need to know what that means. The mitochondria is the site where we make ATP, or energy. We see lysosomes. Lysosomes are going to be the place where we break things down we don't need anymore. So this was your Golgi, packaging and sorting. Rough ER, that's where we made proteins. Smooth ER, that's where we move our fats around. Sometimes the fats even transport through your Golgi. On the outside of your plasma membrane, you may see flagella and you may see cilia. Cilia and flagella are both for movement. You don't see the other structures we talked about, the fimbriae, and pili. We're looking at a eukaryotic cell, so we only see cilia and flagella. So again, I have a slide for you to use so that you can kind of study what the function of all those parts are, and I've given you the important page numbers that you can go back and really look at this information. The last few slides look at physiology. And what we really need to think about with this little section on physiology is how can things move in and out of the cell. So the first thing to consider is movement of water. Movement of water is actually more important for these prokaryotes a lot of times because they are often the organism as a whole. That one cell is the organism. So if that one cell dies because of too much water loss or too much water gain, then the organism's dead. If you think about your body, if one cell doesn't make it, the other cells can be fine. So this water control is very important when we're thinking about prokaryotic cells, or any microbe for that matter. So the best possible situation would be to have an isotonic environment, where the cell, shown here in gray, has the same concentration of water as the liquid surrounding the cell. So you may still see some water movement back and forth, but overall the cell maintains its integrity. Where we can get into trouble if we're a prokaryotic cell is when we have a hypertonic solution or a hypotonic solution. With a hypertonic solution, the concentration of the water is higher inside of the cell than outside of the cell. Okay, so we can say that a different way. The concentration of solute or other stuff is small in the cell, and the concentration of other stuff, the solute, is high outside the cell. So I like to think about and pay attention to the water. To me, that's a lot easier. So inside the cell, if we're 20% stuff, we're 80% water inside the cell. So outside the cell, we're 40% stuff, which means we're 60% water. So looking at the percentages of water, where is there more water? More water in the cell, water is likely to move out. If water moves out of this poor little cell, what's going to happen to the cell? He's going to shrivel up and ultimately die. So that's hypertonic, more water inside. If we look at hypotonic, it's the opposite. You've got more water outside, 90% water outside, 80% water inside. So where's the water going to go? Water's going to rush into the cell, causing the cell to swell. Eventually the cell will lyse, it will burst with too much water on the inside. So why does this matter? Have you ever heard of controlling the amount of microbes present by adding salt? Salt is an example of a solute, it's stuff. So a lot of those canned foods or meats and things like that, to keep bacteria from growing, we'll put salt on them. Well, that high amount of salt makes you have more water inside. So if bacteria are there, with all that water inside, the water will leave, bacteria will die. And so our last thing to consider is transport. There's three major types of transport, which is how do we get things across the plasma membrane. There's simple diffusion. Simple diffusion can occur when you have small, non-polar items trying to pass across the plasma membrane. If they're small and non-polar, they move with the concentration gradient. That is simple diffusion. So in this picture, you see we have more on this side of the cell. So they will move by simple diffusion, slowly beginning to move through the membrane until you get some more on this bottom side of the membrane. This keeps going over time until you have equal amounts on both sides of the membrane. Simple diffusion. with the concentration gradient. Facilitated diffusion is still with the concentration gradient, but you've got a larger or a charged molecule, so it can't move directly through the membrane on its own. It needs a transporter protein. It needs a channel of some sort. So as you can see in this picture, we're still looking at the little green and red shapes, right? Well, they're going to move from the area up top where there's more to where there's less. But instead of going straight through the membrane, they're going through this channel. And then our last type of transport is active transport. Active transport goes against the concentration gradient. So you're going from where you have less to where you already have more. So this is going to use energy. And that's the big key that makes this active transport. Uses energy against the concentration gradient. So as you study the material in this chapter, you really need to take it in segments. Go ahead and learn your history, learn your people, what they did for microbiology, learn the parts of the prokaryotic cell, learn the parts of the eukaryotic cell. Now compare prokaryote to eukaryote. And then finally, understand the movement of water and other items across the plasma membrane.